Welcome back to Digital Learning DC. Myself Dr. Manjula S, working as Assistant Professor of Botany. In today's session, let us study about host, pathogen, symptoms, disease cycle and control measures of coffee rust. Coffee rust is a fungal disease where we can observe rusty mass of fungal spores produced on the coffee plant as a result of pathogen infection, hence the name coffee rust. Coffee rust is the most devastating disease of coffee in the world. This disease was reported on wild coffea species in the Lake Victoria region of East Africa in the year 1861. Originated from Ethiopia, Sudan and Kenya, later spread to Asia and America. This disease is caused by a fungus called Hemilia vastatrix. Hemilia referring to half-moon shaped smooth characteristics of the spores produced by this fungus, whereas vastatrix refers to devastating nature of the disease. As a result of severe infection, this disease is going to cause a devastating loss to the coffee plant. Here we can observe the symptoms of coffee where it is infected by a pathogen, Hemilia vastatrix. Later, as a result of severe infection, the leaves are going to fall off. These are the spores of the fungus Hemilia, which are half-moon shaped and smooth wall spores, hence the name Hemilia, and it is going to cause a devastating loss, hence the name vastatrix. Name has been derived from this region. Then coming to host and pathogen of coffee rust disease. Host is Coffea arabica and Coffea cyniophora. Coming to pathogen, Hemilia vastatrix. This fungus belongs to the class Basidiomycetes under Uridinales family Puccineaceae. Here the clusters of short pedicel bearing dikaryotic spores called iridospores are going to be produced, which are going to protrude through the stomata on the undersurface of the leaf. Occasionally, under dry conditions, I mean to say when there is no rainy season, towards the end of the season, other type of spores called teleospores are produced among the iridospores on older attached leaves. Here we can observe the leaf with the symptoms where it is going to produce under surface of the leaf we can observe number of iridospores which are produced in a clusters. These iridospores on detachment when they fall on the another new host plant, it is going to infect and by producing a germinating hyphae. Then coming to the pathogen of coffee rust, that is Hemilia vastatrix, following karyogamy, that means fusion of the nucleus results in the formation of diploid nucleus, and this diploid nucleus undergo reductional division called meiosis. The teleospores germinate to produce another type of mycelia called basidia, which is otherwise known as basidium, each of which forms 4 haploid basidospores. In the last slide, I explained about the teleospores which are usually produced during unrainy season. These teleospores are usually bicell in nature and they are provided with 2 nucleus. So each cell germinate to produce basidia. On this basidium, 4 basidospores are going to be formed. This basidospore will germinate in vitro. I mean to say when they are on the surface of the basidium only, they start their germination. But it is not known what plant they are going to infect after liberation from the basidia. It is clear that they do not infect the coffee plant. For coffee rust disease, no alternate host is necessary. Hemilia vastatrix can survive and reproduce quite nicely by the formation of iridospores alone. Move on to the symptoms of coffee rust. Let me begin with the initial symptom, that is initially small pale yellow spots appear on the upper surface of the leaf. we can find very small pale yellow color spots on the upper surface of the leaves, where these spots gradually increase in diameter and size and produces mass of yellow color spores called iridospores on the lower surface of the leaf. These powdery lesions on the underside of the leaves are orangish yellow in color or reddish orange in color, hence the name rust disease. They look like a rusty mass, hence the name rust disease. The lesions develop anywhere on the leaf surface. These patches or lesions develop anywhere on the leaf surface, but they usually tend to concentrate around the margins where dew and rain droplets collect. So in the slide we can observe various symptoms like the pale yellow initial symptom, appearance of pale yellow spots which is increase in the size. Under surface of the leaves, it is going to produce a mass of spores called iridospores. It gradually increase in size and produce a number of spores in the particular area, infected area, and these spores usually restricted to margins where dew and raindrops are collected. Continuing with the symptoms of coffee rust, later the centers of the spots, the yellow color spots, eventually dry up and they turn brown in color. They become dry and they brown in color, while the margins will remain yellow. This yellow brown area surrounded by hollow yellow margin lesions continue to expand and produce number of iridospores. Early in the season, the first lesion usually appear on the lowermost leaves. The symptoms usually appear on the lowermost leaves of the plant, and the infection slowly progress upward in the tree, and the all infected leaves soon they will fall off prematurely, leaving a long expanses of twigs which devoid of leaves. The affected trees have significantly lower coffee yield because since the leaves are the photosynthetic area, if the leaves fall off very early, the photosynthesizing capacity of the plant is going to be decreased. As a result of yield of the coffee plant is decreased. Within a few hours, the whole plant is going to die. Then coming to the disease cycle, I already told it is mainly going to produce the spores called iridospores. These spores are responsible for initiating infections. They develop into lesions and produce more and more number of iridospores. When the fungus survive primarily as mycelium in the living tissue of host, this mycelium start infecting the leaves, and as a result of that it is going to produce number of iridospores and the leaf turn brown in color and it will fall off. A few green leaves always persist to the dry season where iridospores can survive for about 6 weeks. In the picture we can observe that this is a iridospore which is going to infect the coffee leaf through small opening or stomatal opening. When it is infecting, it starts producing a vegetative mycelium which is usually intercellular in nature, and as a result of infection it is going to produce in later stage, it is going to produce a knob-like absorbing structure called hostoria. Through this hostoria it will absorb nutrients from the host plant and it will establish very well in the leaf of coffee plant, so that when attained certain stage of maturity, underside of the leaf of coffee plant, this hyphae is going to produce a number of spores called iridospores. Iridospores on detachment, again when they fall on the another fresh plant, they are going to germinate to produce new hyphae of the fungi. So this is taking place during the rainy season. While in unrainy season, at the same place where iridospores are formed, other type of spores called teletospores are going to be formed. These teletospores are bicelled dikaryotic spores which later on produces a mycelium called basidium. This basidium on its surface going to produce 4 basidospores. This basidospores liberate into the soil. Till now it is not identified what plant this basidospores are going to infect. Then coming to the disease cycle of coffee plant, in this picture we can observe the disease cycle in production of spores, where the iridospore, it has the capacity, which is a half moon shaped smooth wall spore. When come in contact with the coffee plant through the openings called stomata, it will enter by producing a germ tube, and it start establishing inside the host cell. The hyphae is both intercellular and intracellular. The hyphae proliferate in between the cells, and they are going to produce certain knob-like structures absorbing organs called ostoria. Through this ostoria, it will absorb food from the host, and it established very well in the host tissue. So after establishment, when it attains certain stage of maturity, it is going to produce a sori, iridosori, in which number of brown color, reddish brown or orangish brown color spores called iridospores, which on detachment, when come in contact with fresh leaf of coffee, it is again have the capacity to infect and produce hyphae. So this is the normal cycle which is taking place. During unrainy season, at the same point where iridosori are developed, during unrainy season, a tilitosori is going to be formed, which is going to form another type of spores called tilitospores. These tilitospores are bicell structures with a dikaryotic nucleus. They later develop to form basidium, which is nothing but a promycelium of the fungus. On the basidium, four number of sterigmata, which are the bottle shaped structures, are going to be formed, which cut down 4 basidospores, which released into the soil. So in some cases, the tilitospore also have the capacity to infect the coffee plant. So as a result of both iridospore and tilitospore infection, the coffee plant, different leaves of the plant get infected, and as a result of severe infection, the leaves will wither off, resulting in decrease in the yield of a coffee plant. Let us move on to spore dispersal, germination and infection of Emilia vastatrix. Emilia vastatrix fungus is going to produce a spore called iridospores, which can be dispersed by both wind, blowing wind, and also rain splashes. A small amount of iridospores also carried by certain insects called vectors. Certain vectors, they act as a carriers. They also disperse the spores, which includes thrips, flies, wasps, other insects. Iridospores germinate only in the presence of water. Iridospore require water for germination. The infection is usually occurs only during the rainy season. That's why the rainy season is the preferable environment, preferable season for the germination of iridospore. They enter the host tissue through small opening called stomata. So it takes 10 to 14 days from the infection point for a new iridia to develop, inside which a number of iridospores are going to be formed. The rust lesions continue to enlarge over a period of 2 to 3 weeks. A single lesion will produce 4 to 6 crop of spores, releasing about 300,000 iridospores over a period of 3 to 5 months. Coming to control measures. Control measure includes the following management strategies like field sanitation. So it includes collecting and destroying the infected plant parts. So as I already told, the main part which is going to be infected by the pathogen is the leaves. So we have to, as soon as we found any small symptoms, that is pale yellow color patches, we have to remove that, we have to separate that leaf from the main coffee plant and destroy them. The infected plant part is one of the methods to control the disease. Collecting and destroying the infected plant part is one of the methods to control the disease. Then coming to the second chemical chemical treatment method, that is spraying of copper fungicide. Since the Emilia vastatrix is a type of fungus, we can use certain chemicals which are used to kill the fungi, like body ox mixture at 0.5% at proper intervals is an effective control measure. Along with body ox mixture, certain other fungicide like dithiocarbamate, which is a sulfurous compound. They are also effective in controlling the disease. Proper maintenance of canopy cover. Canopy is nothing but the cover over the top of the coffee plant. It is also one of the control measures since it prevents the dew formation. It is also one of the method to control the disease incidence. So using resistant variety of coffee plants can reduce the disease incidence and control the disease. I am going to sum up the session with the following points. Coffee rust is the important devastating disease of coffee plants, where coffee is a most important commercial crop which is generally used as a beverage throughout the world. The disease coffee rust is going to cause it is going to cause less yield in the coffee plant, which is caused by a fungus Emilia vastatrix, and the fungus mostly infects leaf blades, where the photosynthesis rate is going to be stopped. As a result of that, yield is also reduced. It is very rarely attacks the berries. Symptoms appear in the form of yellow spots, gradually increase in size and produce orange powdery mass in the later stage, which is nothing but the iridosauri enclosing number of iridospores on the lower side of the leaf. As disease advances, leaves start dry up and they fall off very early, prematurely, so decrease in the yield of berries. Fungus reproduce and survive from season to season in iridial stage, where iridospores are going to be formed. They spread through blowing wind and rain splashes. The disease can be managed by field sanitation, spraying fungicides like body ox mixture, maintaining proper canopy, and also by using disease resistant coffee varieties. With this, I am going to sum up the session. For further details about the current topic, you can refer the following references. Thank you.